It's a full screen, right?
Correct. All good.
Okay, great. So thank you, Jerry. Thank you, Carlos and Elena, and all the organizing committee for organizing this committee dedicated to for students, international students. So it's a real pleasure for me to be there, and for thank you for giving me the opportunity also to present my research. So I will start so that I will try to respect the time slot you gave me. And yeah, please feel free to interrupt me if you have any question during the talk, or I mean, it's up to you. I don't know if you prefer to ask me the question at the end. For me, everything is fine. Um, so today I'm going to talk about volcanoes and how they can potentially impact our health over time. And just before to start, I just would like to give you a brief overview of why we've decided to work on volcanoes and the link with health. As you can see on this map,
I'm
uh,
sorry, Dr. Seauzéat, we
yeah,
lost your sharing. May you please share your screen once more, please?
oh yeah, sure.
Thank you.
Is it working now?
Yes, it's working. Thank you.
Okay, I don't know. I didn't do anything, so. Uh, but can you see my laser?
Yes, we
Yes,
can.
we do.
Okay, perfect. So yeah, so I was saying that volcanoes and many active volcanoes that are shown here by the red triangles are located everywhere into the world. So they spread all over the continents, and most of these active volcanoes are located in very dense populated areas that are shown here in dark green. And today, just to give you an idea, it is estimated that more than 10% of the world population is living within 100 kilometers of an active volcanoes. And so most of these people are living in developing countries because most of the active volcanoes are located in developing countries. And so these people are frequently exposed to volcanic products that are released by volcano either during or after an eruption. And one of the main products released by volcanoes are volcanic ash, with more than 1 million cubic meter of ash released into the Earth's atmosphere every month. And yeah, just I would like to say, because most of the time when I'm talking about volcanic ash, people are thinking of very fine silicate particles, fine ash, mainly lower than 10 micron, what we call the PM10. But by definition, volcanic ash is a very complex and heterogeneous mixture of particles, including everything that is lower than 2 millimeters. So it's not only the fine PM10 particles. And so volcanic ash, so as I said, so it's a complex mixture of particles with different forms, size, textures, chemical composition, for example. And so people can be exposed to this volcanic ash during an eruption. And so in that case, we talk about acute exposure. And so the exposure can occur by multiple pathways, including by the nose. We are talking about nasal inhalation, but also by the mouth, either by inhalation, but also by oral ingestion. And it is also important to keep in mind that, okay, people are exposed during an eruption, but after an eruption, volcanic ash that has been released within the atmosphere will, at some point, at some time, will sediment deposit on the ground. And here you have an example. So it was after the Chitin volcanic eruption of the Chitin volcano in Chile in 2008, and we can see that the soils it completely covered by volcanic ash. So we have ash blanket. And then, either by wind or human activity, this volcanic ash can be resuspended, such as, for example, when people are cleaning or cutting the grass, such as, for example, here at the Soufrière Hills in Montserrat. And also, ash can also be resuspended, for example, by car traffics. And so, and this resuspension process can occur several months or years after an eruption. And so people, in that case, we are talking about, chronic exposure. So, and so this exposure to volcanic ash, but not only, also to other volcanic products, including ash, but also contaminated water and food, have been shown to induce significant health effects, such as, for example, ocular effects, cardiovascular, dental and skeletal, dermal and respiratory effects. But if we focus only on the health effects induced only by ash exposure, so far, the studies mainly reports respiratory disorders. And so the question is, why? Is it because volcanic ash exposure only induce respiratory alterations, or is it because people only look at respiratory disorders? And just to give you an example regarding these respiratory disorders, so this is based on epidemiological studies based on humans, but also in vitro on cells and in vivo studies conducted with animals, mainly mice. And so here you have two examples just to illustrate what I'm saying. So on the bottom, you have a diagram showing the number of the visits to the emergency for several respiratory diseases, such as, for example, hyperventilation, cough, or acute bronchitis, as a function of time. And we can see that when we have a volcanic eruption that is shown here by the red arrow, we can see that we significantly increase the number of visits to the emergency. And this is associated with a significant increase in the number of TSP, which is the total suspended particles into the air. And so this significant increase was directly related and associated to the increase of volcanic ash released by the volcanic eruption. And then here you have another example. So it's from an in vivo studies. So in these studies, the author exposed fisher rats to volcanic ash with intratracheal ingestion. And they show that, so here you have an image of the lung. And it was after several months of exposure, the rats tend to develop granulomatous lesion into the lung, which is a sign of a pulmonary inflammation. And so I'm not gonna detail and list all the studies that show and demonstrated the link with respiratory disorders, but if I try just to summarize what we know today, is that so acute exposure to very fine volcanic ash, what we call the PM10, have been shown to induce respiratory health effects, and this is associated with adverse lung injuries, and this is something that has been shown mainly with in vivo studies, and this is also associated with a weak pro-inflammatory response, as highlighted by in vitro studies, so working with mainly lung cells. But the problem is that for now, we don't really know what are the main toxicity parameters. So several hypotheses has been suggested, for example, particle size, morphology, or interaction with pollutants. Another question is that we have, as I said, we have quite information on what's happening to the lung and the respiratory system, but we have very limited information on what's happening to the rest of the organism. Do volcanic ash exposure affect other reservoirs, other organs? We don't know yet. And then the last question is, what are the main chronic health effects? Do we have similar effects that are induced by acute exposure or not? And so to try to answer this question, we've decided to adopt an interdisciplinary in vivo approach. So briefly, the idea was to expose mice, isogenic mice, black cysts, to volcanic ash. Okay, sorry. And because not all volcanic ash has the same toxicity, we decided to work with different type of ash from several distinct environments. We worked with ash from La Soufrière de Guadeloupe, with ash coming from the Ubinas volcano, so an andesitic volcano from Peru, and from the Tarogaite volcano in Spain, which is with a more basaltic composition. And so we try to work with as much as we can with fresh ash sample. except from La Soufrière de Guadeloupe ash, because we didn't have any access to fresh ash because the Guadeloupe volcano didn't erupt in the last years. So to face off this problem, we've decided to work with, we've collected pieces of rocks from the dome, from the volcanic dome that we crushed, we sieved to try to reproduce what we call an artificial volcanic ash with a median grain size around 100 micron. And as you can see, it also includes a significant amount of fine ash, lower than 10 micron. And so we exposed mice to this volcanic ash over several months, till 2 months, to try to mimic chronic exposure. Just to give you an idea, 2 months for mice, it's about equivalent to 5 years for a human. And after the exposure, we've collected several organs, including the lung, but not only. We also collected testes, kidney, liver, heart, brain, but also blood, urine, and the feces. And then we measure a bunch of parameters, including metallomic parameters. So that is to say everything that is related to metals. This can include the concentrations, but not only, also the isotopic composition, and I will talk about that later. This can also include the speciation. And we also look at morphological parameters, histological data, and we also couple that with what we call the omics measurements, including metabolomics, proteomics, and transcriptomics, all these measurements being organs specific. And so now let's move to the results. So of course, I won't have time in 25 minutes to show you the result for all the organs. So I'm going to focus on one specific one being the testis. So here you have a principal component analysis diagram. So, and in this diagram, so you have the red circles show the testes collected on 5-month-old mice, either, so in light green, not exposed to volcanic ash, in dark green, exposed over 1 month to volcanic ash, and the same, the square dots represents mice exposed, so still the same color code, light green, mice not exposed to volcanic ash, and dark green, mice exposed to volcanic ash over 2 months. And what we can see on this diagram, on this PCA diagram, is that we have, mice exposed to volcanic ash are characterized by the significant metallome deregulation. And this is something that is maybe much more visible on this biplot diagram, showing the copper concentration as a function of the iron concentration still in the testis. And we can see that, so here we have mice not exposed to volcanic ash, and mice exposed to volcanic ash tend to have much higher content in copper and iron content. So meaning that we have a significant metal accumulation within the testis associated with the exposure to volcanic ash, and we have the same tendency either after 1 or 2 months of exposure. And then we demonstrated that this metal accumulation in the testis is associated with significant testicular alteration, such as, for example, so it's associated with a decrease. So sorry, in this presentation, it will always be the same color code. So in light green, it will be mice not exposed to volcanic ash, what we call the control, and in dark green, mice exposed to volcanic ash. And so we can see that the metal accumulation observed in the testis is associated with a decrease of the number of spermatozoa in the tail and the head of the epididymis, which is the space where the spermatozoa tend to be stored before being excreted out of the organism. We also observe histological deregulation, such as, for example, a decrease of the epithelium thickness ratio. So just for the one that are not familiar with what is the testicular epithelium. So here you have an illustration of a testis. A testis is composed of what we call seminiferous tubules. And here you have a thin section of 1 tubules. And as shown here by the red arrow, this is the epithelium, the testicular epithelium space where the germ cells are located. The germ cells being the cells that will form the spermatozoa. And so in mice exposed to volcanic ash, we have a significant decrease of this testicular epithelium thickness. And we also observed histological deregulation, such as, for example, epithelial desquamation, and also interstitial inflammation and abnormal germ cell location, as shown here by the red arrow. And then we also perform immunohistochemical analysis. So I'm not going to enter into too much detail, but the main information is that we observed, so in mice exposed to volcanic ash, still illustrated here in dark green, we observe a significant decrease of the number of spermatids. And the spermatid is the precursor of the spermatozoa. And so all this information, all together, these results demonstrate that, suggest that ash exposure can affect the spermatogenesis process, which is this process, the biological process leading to the formation by several steps of meiosis and mitosis to the formation of spermatozoa. And so this question about, okay, if ash affects the spermatogenesis process, do volcanic ash exposure affect the fertility? And so to answer this question, we've decided to perform a reproduction test. So for that, we exposed mice, male mice, to volcanic ash over 2 months of exposure. And then we mated this mice with female, not exposed to volcanic ash previously. And then we look at the number of pups per liter. And what we observed is that female mated with male previously exposed to volcanic ash have a lower number of pups per liter compared to the mice that were not exposed to volcanic ash. And so this suggests that volcanic ash might induce hypofertility, might account for hypofertility. And so the next question was, okay, if ash induce hypofertility, what are the mechanisms accounting for this ash-related hypofertility? And so the processes are likely multiple and complex. One of the main one is likely a liver testis crosstalk. So we've demonstrated that ash can also affect the liver, and liver deregulation has been shown to alter fertility, but I'm not going to explain this today. I will only focus on a second process, that is the oxidative stress. So by definition, oxidative stress is the imbalance between the production of oxidants, what we call the ROS, for the reactive oxygen species that are very unstable species, aggressive species, and antioxidant defense and this may result in damage to biological system. And it has been shown that oxidative stress is involved in a wide range of diseases, such as, for example, testicular injuries. And so when the oxidative stress increase, it means that we increase a reactive oxygen species, and by opposition, you tend to decrease the number of antioxidant species. And in the organism, the main antioxidant enzymes are the SOD for the superoxide dismutase, the GPX for the glutathione peroxidase, and CAT for the catalase. And so in the case of volcanic ash exposure, still I'm going to focus only on the testis. What is shown on this diagram, so here you have is represented the iron concentration as a function of the expression of the gene coding for the GPX3 enzymes. So just to remind you, it's one of the main antioxidant enzymes. And what we can see is that mice exposed over 2 months of exposure are marked by a significant increase of the iron concentration, as I said previously, and this correlates with the downregulation of the GPX3 expression. So meaning, and this likely reflect an increase of the oxidative stress. And this increase in oxidative stress is likely not only confined to the testis, it's probably expressed and something general that can be observed at the entire body scale. And here you have an example. This is something omnipresent, in the liver. And here you have an example. So here you have, in this diagram, is shown the expression of several genes coding for all the main antioxidant enzymes, SON 1 to 3, GPX 1 to 4, and the catalase. And what we can see is that in the liver, except for the SOD 1, we have a significant decrease for most of these, or at least a tendency to decrease for all these antioxidant enzymes, which is a sign of an increase of the oxidative stress. And so we suggested that this oxidative stress that is observed at the entire body scale might account for hypofertility. And then just to finish, that's where isotopes, because if you remember in my title, I was talking about isotopic composition. And so that's where isotopes can be helpful and come into play. So just because I'm not sure that everyone is familiar with what is an isotopes and how do we measure isotopic composition. So just by definition, so isotopes are forms of the same chemical element with equal numbers of protons at the atomic scale, but different number of neutrons. So this means that for one specific chemical element, such as, for example, the copper, two isotopes of the same chemical element will have different masses, and so will have different physical-chemical properties. And so they will behave differently during the biological processes. And so just to give you an example, if you look at copper, so copper is a chemical element with two stable isotopes, the light one, the 63, that is much more abundant than the heavy one. And so the isotopic composition is just the ratio of these two stable isotopes normalized to a reference material. And basically it's multiplied by 1,000 because these variations are very small. And until recently, isotopic composition were only measure in earth sciences, but until I will say 10 years ago, they were transferred to the medical field, and now we are talking about the isotope metallomics. I don't know if you heard about it. And just to say, so it has been shown that measuring isotopic composition applied to the medical field have been shown to bring additional information compared to the concentration. It has been shown that isotopic variation are linked to biological processes, their regulation, and might be very helpful and appear as promising future markers of diseases. And yeah, just to give you an example, for example, if you look on the bottom left diagram, is shown the copper isotopic composition in human serum. And what is shown is that so healthy people in green tend to have a much higher isotopic composition, copper isotopic composition, compared to people having a hepatic diseases. And so in this study, the author suggested that measuring the copper isotopic composition in the serum might help to diagnose the development, the onset of the hepatic diseases. And so in our case, so here again, I'm going to focus on the testis. So here is shown the isotopic composition of three isotopic system, the copper isotopic composition, the zinc isotopic composition, and the iron isotopic composition measured in the testis of mice controlled in light green, mice exposed to volcanic ash over 2 months in dark green. And what is shown on this diagram is that after 2 months of exposure to volcanic ash, we have a significant increase of the copper isotopic composition. Zinc, which is a non-redox sensitive isotopic system, remains stable. And we also observe a significant decrease of the iron isotopic composition. And then we also observe that these isotopic shifts are associated with the downregulation of all the main antioxidant enzymes. And so here you have several examples. So here is shown the copper isotopic composition in the testis as a function of the gene expression of the GPX3 gene expression in the testis. Same when we look at the superoxide dismutase. And same when we look at the iron isotopic composition, we have significant correlation. And this is something that is true when we look at the transcriptomic scale. So we are looking at the transcript, and the same when you are looking at the protein activity, so at the proteomic level. And so this means that this suggests that redox sensitive isotope ratio might be reliable markers of oxidative stress. And so just to summarize briefly, what we show is that volcanic ash are enriched in metals or potential toxic elements. You can call it as you want. These metals, once absorbed by an organism, can released. So these metals can either directly account for the formation to reactive oxygen species, such as, for example, via the Fenton-like reaction, and or can downregulate antioxidant enzymes fighting against these reactive oxygen species. In both cases, this increases the oxidative stress, and this oxidative stress might account for testicular disorders that in turn might induce hypofertility. And then we also demonstrated that measuring redox-sensitive isotopes, such as, for example, copper or iron isotopic variation ratios, can be promising and help to diagnose oxidative stress and then prevent hypofertility in volcanic environments. And so I will stop here. I'd be happy to answer your question. And oh yeah, I forgot. So we are hiring. So we are looking for a 2 years postdoc funded by the French National Research Agency, a postdoc that will be working in isotope geochemistry, specifically on redox-sensitive isotopic analysis and their application in volcanic health hazard. So yeah, if you have expertise in isotopic analysis and if you have notion of molecular biology, please feel free to contact us. And or if you know someone that might be interested, please feel free to spread the word. And yeah, so I will stop here. I hope you can still hear me. Maybe I can stop sharing my screen. Can you hear me?
Yes, we can.
Okay.
Jerry, are you?
So Lucie, I will interrupt here to say thank you very much for this excellent presentation. Yes. I may ask one question, one curiosity, please. You started you're talking about using how to discriminate between acute and chronic toxicity, let's say, and you are showing that you can do by isotopic analysis, you can do such discrimination. Can you elaborate a little bit? Do you find already any differences between an acute exposure and chronic exposure? Do you see differences in the isotopic signatures? Is that it?
Okay. Can you still hear me?
Yes.
Yeah. No, okay. So maybe it was not enough clear. I'm not saying that I can discriminate between chronic and acute exposure with isotopes. I'm just saying that measuring isotopic composition might give us information about what's happening, what are the mechanisms that are developed, and what are the processes developing, and that might account for diseases developing in volcanic environment. But I didn't focus on acute exposure. In my experience, I only try to mimic chronic exposure. So, but I'm not saying that isotopes cannot give us information about chronic exposure, but probably, I mean, I don't think that's the same mechanism that are developing in the case of acute and chronic exposure.
Okay, thank you.
I don't know if it was clear.
Just a curiosity because it's not my field of expertise. So excellent talk. Thank you very much, Lucie.
Thank you.
Yes, thank you, Lucie. There's a question in the chat window. Ariadne is asking if significant effects of ash exposure were noticed on organs too in the environment.
Oh, I'm sorry. Can you repeat?
Hi, can you hear me?
I didn't hear what you said. But maybe I can read directly into the chat.
Did you? Okay, yes, in the chat, yes.
or let me help you.
Yeah, sure.
Professor Arguelak is asking, do you observe significant effects of ash exposure on other organs too in your experiments?
Yeah. So it's different. So after 1 month of, because we perform we perform assays. So we expose mice over 1, and then we increase the time of exposure till 2 months of exposure. So after 1 month of exposure, it's mainly the liver and then the blood that is affected, and just a bit the testes. But we didn't observe significant variation for the other organs, but I'm talking about mainly metallomic variation. So I'm not saying that we, for example, for the lung, that we didn't look at, for example, inflammatory markers. So I'm not saying that I'm only talking about metallomic variation. And after 2 months of exposure, we observe significant variation for most of the organs. So clearly there is something happening between 1 and 2 months of exposure.
Okay, thank you very much. Can you hear me?
Jerry, it seems you have some
Hello?
connection problem, I think. So thank you, Dr. Seauzéat.
Can you hear me now?
Yes, we can, Jerry.
Yes, thank you, Lucie. I think time for just one more question. There's one more question in the chat. Professor used in research.
I'm sorry, can you repeat? I cannot hear you well.
Professor Demetria is asking if mice are out to use the research.
Sorry, I didn't hear what you said.
Sorry, Jerry, it seems you have connection problems. So
I can ask the question just really quickly because we need to keep going. So the question is it allowed to use mice in research?
Yeah. Yes, of course it is allowed. Otherwise, we won't be working with mice. It's highly regulated with European regulation, and so we have to write specific proposal, and that it has been to be validated by ethical committees. But yeah, a lot of restriction, but yeah, it is possible. But we are encouraged to work with also to develop working with cells with in vitro in parallel to avoid working with too much mice. But
okay. Thank you, Lucie, for this excellent talk, and thank you for being here today, giving this tremendous view of your work to all of us, and all the success for you. Thank you very much.
thank you.